Genetic disorders can be classified into 4 types. Single gene disorder, multifactorial disorder, chromosomal disorder, and disorder with somatic mutation. In genetics, a trait is a characteristic such as hair color or blood type that can be passed from one generation to another. Over 11,000 traits in humans. 5,000 of them are clinically significant disorders. They exhibit single gene inheritance. They are primarily disorders of pediatric age range. Less than 10% manifest after puberty. Before we move on to talk about inheritance, first, I need to introduce you some genetic terms. The physical location of a gene on a chromosome is called a locus. Variation in the DNA sequence of a gene produce different alleles at one locus. Each pair of alleles represent the genotype of a specific gene. In diploid somatic cells, a genotype may be homozygous if the individual has the same allele on both homologs at that locus. And genotype could be heterozygous if the individual has 2 different alleles on the 2 homologs. Phenotype is the observable and measurable physical characteristics of an organism. A phenotype expressed in heterozygotes is classified as dominant. A phenotype expressed only in homozygotes or hemizygotes for X-linked traits is recessive. Recurrent risk is the probability that the offspring of a couple will express a genetic disease. It is important to remember that each reproductive event is statistically independent of all previous events. Therefore, the recurrence risk remains the same regardless of the number of previously affected or unaffected offspring. A pedigree describes a patient's family history. First affected individual to be identified in the family is called the proband, as indicated by an arrow. Here, we use a square to represent a male and a circle to represent a female. A filled square or circle represents an affected individual. A symbol with a slash represents an individual who is deceased. And this symbol with a note SB represents stillborn. And these are symbols for dizygotic twins and monozygotic twins. Today, we will cover 7 topics. We will first look at autosomal dominant disorders. Here is a pedigree showing the transmission of an autosomal dominant disorder. An autosomal dominant disorder is observed in multiple generations of a pedigree. Skip generations are typically not seen. Both male and female are affected in roughly equal frequencies. Among 6,000 known Mendelian traits, more than half are autosomal dominant. Here are some well-known autosomal dominant disorders. Except neurofibromatosis type 1 or NF1, all of these disorders have been covered before. Next, we will take a closer look at NF1. NF1 is caused by mutation in neurofibromin, a gene involved in the RAS pathway. NF1 is characterized by the growth of multiple benign tumors in the skin. They are also called neurofibromas. Presence of multiple flat, irregular pigment skin lesions known as cafe au lait spots. The growth of small benign tumors on the iris of the eye. If the capital A represents the dominant mutant allele and lowercase a represents the recessive normal allele, the various combinations of the gametes can be represented in a square. It is called Punnet's square. Any child born to a person affected with a dominant trait has a 50% of chance of inheriting it and being affected. Autosomal dominant disorders may have some variations. First variation is new mutation. As shown in this pedigree for NF1, the proband's parents and their parents are unaffected. The mutation this man has must be originated as a new mutation, most likely from his father. New dominant mutations in some instances have been associated with the increased age of the father. Second situation in which variation occurs is sex-limited phenotype in autosomal disease. As shown in this pedigree, an autosomal dominant trait is passed from a man to his boys, but not to girls. An example for sex-limited phenotype is familial testotoxicosis, an autosomal dominant disorder. Affected boys developed secondary sexual characteristics and undergo an adolescent growth spurt at about 4 years of age. In some families, it is caused by mutation in the gene coding for luteinizing hormone receptor, located on chromosome 2. Most of the autosomal dominant disorders occur in the heterozygous state. Some autosomal dominant disorders appear in homozygous state. In some cases, such as achondroplasia and familial hypercholesterolemia, individuals in homozygous state are more severely affected than heterozygotes. On the other hand, in some other cases, such as Huntington disease and myotonic dystrophy, homozygous individuals are not more severely affected than heterozygotes. Now, let's move to part 2. We will look at autosomal recessive inheritance. Here are some well-known disorders that follow autosomal recessive inheritance. We have covered all of them before. People with autosomal recessive inheritance inherit one copy of the disease-causing allele from each parent. Autosomal recessive diseases are typically seen in only one generation of pedigree, as shown here. Because these genes are located on autosomes, male and females are affected in roughly equal frequencies. Most commonly, a homozygote is produced by the union of two heterozygous parents. Again, we use Punnet's square to determine the recurrence risk. Here, capital A represents normal allele. A lowercase a represents the disease allele. The recurrence risk for offspring to be homozygous for the disease allele is 1 in 4, or 25%. Consanguinity is the mating of related individuals. In MBME exams, consanguinity is often seen in recessive pedigrees. MBME requires that students know how to determine recurrence risk under two situations. First situation involves an unborn child from consanguinity. For example, if in the first generation, either parent carries a disease gene, such as FCFTR mutant, how much probability will the child from union of first cousin or second cousin be affected? Here, I provide you answers. Detailed calculation should be done by students as homework. The second situation involves an individual whose phenotype is known. For example, for individual 4-1 in this pedigree, how much is his risk to be a carrier? Because 4-1 is phenotypically normal, his phenotype is known. When we use Punnet's square, we should rule out the possibility of this man being homozygous for the recessive allele, as shown here. As a result, there are only 3 possibilities. Out of these 3, 2 corresponds to carrier. Therefore, the risk of being a carrier of this man is 2/3. Similar to autosomal dominant inheritance, autosomal recessive inheritance also has variation. A prominent variation is pseudodominance. If an individual who is homozygous for an autosomal recessive disorder, as shown here, marries a carrier of the same disorder, their children have 50% chance of being affected. Such a pedigree exhibits pseudodominance. Now, let's move to part 3. We will look at X-linked recessive inheritance. This type of diseases are seen much more commonly in males than in females. Skip generation are commonly seen. Male-to-male transmission is not seen in X-linked recessive inheritance. This information is summarized in this slide. Most common X-linked recessive diseases tested in the USMLE are shown here. Wiskott-Edridge syndrome is caused by mutation in WASP protein, a regulator of actin cytoskeleton. Bruton A-gamma globulinemia is caused by mutation in a tyrosine kinase. Altogether, this disease can be summarized in one mnemonic. Obviously, female will often give her boys her X-linked disorders. When an affected male is mated with a normal female, all the daughters will be heterozygous carrier. All the sons will be hemizygous normal. When a normal male mated with a carrier female, a half of the son will be affected and half of the daughters would be carrier. There are some variations in the X-linked recessive disorders. The most important variation is X inactivation. A normal male has an X chromosome and a Y chromosome. A normal female has two X chromosomes. An X chromosome carries far more genes than a Y chromosome. A mechanism must exist to equalize the amount of proteins encoded by X chromosomes in males and females. This mechanism is called X inactivation. It occurs in the blastocyst during the development of female embryos. In a female embryo, at blastocyst stage, either paternal chromosome is inactivated and becomes a bar body, or maternal chromosome is inactivated. The choice of paternal versus maternal is random, but once it is chosen, it stays throughout the development. Therefore, cells with multiple X chromosomes, all but one, is inactivated during embryogenesis. In humans with more than one X chromosome, the number of bar bodies at interphase is always one fewer than the total number of X chromosomes. For example, this cell has 3 bar bodies. It has 4 X chromosomes. Several mechanisms work together in X inactivation. The primary mechanism involves cyst gene. The cyst gene encodes RNA that codes the chromosome, helping produce its inactivation. Methylation of gene regions on the X chromosome to decrease gene expression. Condensation of heterochromatin to further make the chromosome inactive. A woman who is a carrier for a disease gene on the X chromosome may have the disease when most of the X chromosome carrying the normal allele have been inactivated by X inactivation. Such females are called manifesting heterozygotes because they usually have at least a small population of active X chromosome carrying the normal allele. Their disease expression is typically milder than that of hemizygous male. Now, let's move to part 4. We will look at X-linked dominant inheritance. As in X-linked recessive inheritance, male-to-male transmission is not seen. Different from X-linked recessive inheritance, heterozygous females are affected. Because females have 2 X chromosomes and males have only 1, X-linked dominant disease are seen about twice as often in females as in males. No generation skipped. When a male is affected, none of his sons will be affected, but all of his daughters have the disease. Here are diseases that follow X-linked dominant inheritance. We will look at fragile X syndrome in the next lecture. To determine recurrence risk, we will use Punnett square again. When an affected male is mated with a normal female, none of his sons are affected, but all of his daughters are affected. If the sex of the fetus is unknown, the recurrence risk is 50%. When a normal male is mated with a heterozygous affected female, 50% of children are affected.
Now, let's move to part 5. We will look at Y-linked inheritance.
In Y-linked inheritance, also called holangic inheritance, only males are affected. An affected male transmits Y-linked traits to all his sons, but to none of his daughters, as shown here. Because Y chromosome is very small, only a few traits are Y-linked. Here are some examples. One is hearing impairment. Second one is HY histocompatibility antigen and genes involved in spermatogenesis. As you have seen from previous cytogenetic lecture, there are two pseudo-autosomal regions on the Y chromosome, here and here. The pseudo-autosomal region creates a variation in Y-linked or X-linked disorders, which is called pseudo-autosomal inheritance. An example is dyschondrosteosis. It is caused by mutation in SOX gene, either on the X or on the Y chromosome. Especially the SOX gene escapes X inactivation. People with this condition have short stature and deformed arms. As shown in this pedigree, this condition can be transmitted from male to male and male to female in a manner similar to autosomal dominant inheritance.
Now, let's move to part 6. We will look at phenotypic variability. Penetrance, expressivity, and pleiotropy.
The differences in expression or phenotype can often lead to difficulties in diagnosis and pedigree interpretation. There are three distinct ways in which such differences in expression can occur. One is reduced penetrance. Second is variable expressivity. And the third, pleiotropy. Penetrance is the probability that a gene will have any phenotypic expression at all. Expressivity is the severity of expression of the phenotype. For example, some people with NF1 have only cafe au lait spots, freckles on the axillary skins. Others may have tumors in the spinal cord. Pleiotropy refers to the phenomenon that one mutant gene produces several different phenotypes. For example, mutation in EYA1 gene cause abnormal ear development. They also cause cysts in the neck and renal dysplasia.
Finally, let's move to part 7. We will look at genetic variability. This is also called genetic heterogeneity.
Some disorders may have similar phenotypes but are caused by different genotypes. This is called genetic heterogeneity. Genetic heterogeneity may result from mutations at different loci. This is called locus heterogeneity, or different mutations at the same locus. This is called allelic heterogeneity, or both. Locus heterogeneity can be demonstrated in 3 disorders. One is retinitis pigmentosa. People with this condition have photoreceptor degeneration. It has been known to occur in autosomal dominant, autosomal recessive, and X-linked forms, indicating mutation in different genes can cause this condition. Second disorder is Ehlers-Danlos syndrome that we have covered before. It has been shown that there are more than 10 different loci associated with this disorder. Third condition is congenital deafness. Results from mating in the first 2 generations indicate congenital deafness follows autosomal recessive inheritance. Results from mating in the 3rd generation are different from what is expected from autosomal recessive inheritance. The reason why this has happened is because woman 37 carries different disease-causing genes compared with man 39. As a result, their children are all double heterozygotes. Allelic heterogeneity can be demonstrated using cystic fibrosis. For example, patient A has a missense mutation, patient B has a nonsense mutation, patient C and D have a frame shift mutation, patient E has a small deletion. Different alleles may result in different clinical presentations. Second example is Rett gene. Some mutations in the Rett gene cause Hirschsprung's disease, as shown here. Other mutations in the same gene cause MAN2A and MAN2B. They stand for multiple endocrine neoplasia type 2A and 2B. A third group of mutation in the Rett gene causes both Hirschsprung disease and MAN2A and MAN2B.